Okay, hi everybody, welcome back. I hope you had a good lunch. And we're just going to jump right in and try to still make up a little bit of time. I'm super excited for our next session. Again, we just have some more amazing CTNNB1 moms. Maybe next year we're going to have some dadvocates in the mix here. This talk is titled The Journey of CTNNB1 Patient Advocacy Orgs Leading Therapy Development, and talking a little bit about our collaboration together. So I'm so pleased to introduce Emily Amerson. She's a CTNNB1 mom, of course, and the president of CTNN Connect and Cure in USA. And then afterwards, we're going to also be hearing from Dr. Ana Gonzalez Hernandez, who's a mom and vice president of the CTNNB1 Foundation. So hi, Emily.
Hello, can everyone hear me?
Yes.
Yay, okay. Thank you all for the hard work that went into organizing this conference and the privilege of being able to present today. As Effie said, I'm Emily Amerson. I'm the president of the CTNNB1 Connect and Cure organization in the US. I'm sad not to be there with you in person, but I'm going to talk on the screen, and then Anna, who is there, will pick up from there. You would probably laugh seeing us organize this together across time zones and with little babies attached to us, but it's been a pleasure to work with her, and we hope that you enjoy. So first, we're going to start off with some numbers. So today we have 8 years of community in the CTNNB1 world. There are 5 patient organizations. We have about 430 patients with known diagnoses. There has been over $3 million raised in donations to support research, almost $1 million in government grants. There are 8 patient-induced cell models that have been generated, 6 animal models, and 6 ongoing therapy programs. So this talk really just comes from a paper that we have all worked on together. You can see the authors here. It's been a pleasure working with all of them. And this is just kind of a highlight of the information that we hope to share with you in that publication soon. And it has been submitted. Okay, so just to walk you through kind of the history of our community so far. In 2016, that's when we first gathered together in a Facebook group. That is also the year that CTNNB1 was added to the Simon Searchlight study. In 2017, the CTNNB1 Syndrome Awareness Worldwide organization was created called CSAW. And then after that, Advancing CTNNB1 Cures and Treatments called ACT was created to focus on research. The first CTNNB1 conference was hosted online in 2020. And then 2021 was a big year. That's when the CTNNB1 Foundation was created to support gene-related treatments. They began their genotype-phenotype study. That's also when the CTNNB1 organizations in Spain and Italy were founded. And then moving into 2022, in the US, we had those 2 organizations, CSAW and ACT, and that's when they officially decided to merge into one, which is now what we call CTNNB1 Connect and Cure. Okay, and then 2023, last year, was another big year for us. That's when France joined us as an organization. That was when the first CTNNB1 conference in person happened in Madrid, for those of you who were there. And then right after that, we had the first US-based in-person event in June in New Jersey. And that's where we actually had our first in-person evaluations of CTNNB1 patients. Later that year, we started the manufacturing process for the gene therapy program under CTNNB1 Foundation. And then this year, as you all know, you're there to participate in the second International CTNNB1 conference with in-person evaluations for the International Natural History Study. Okay, so as we were coming up with this paper to give a big picture of what we have done so far in the community, what the community has built, we came up with these 4 big pillars. First is understanding the disease. So systematic review of the published cases, and 5 natural history studies to understand the disease. Oops, I'm sorry, I'm missing my notes here. Okay, and then developing preclinical models. There are currently 8 patient-derived iPSC cells available to researchers worldwide, and 1 whole body loss of function mouse model. Next, one thing that's really important to us is building community and collaboration. So not only do we have the Facebook groups, we're also hosting conferences and really focusing on connecting researchers around the world to promote science and not duplicate efforts. And then obviously supporting research is at the front of everything. And so we have disease mechanism studies, and we have those 6 ongoing therapeutic programs that we're really excited about. This next slide is just a visual of really how broad our network is. It has in yellow the patient advocacy organizations, and then in blue are the scientific networks, all working for our kids. On the right is the number of patients in each country, and that's from the Facebook Family Connections survey. Okay, so 2 observational studies that we mentioned in the paper that we worked on were Simon Searchlight and the genotype-phenotype correlation study. First, Simon Searchlight is an online international study. Right now, there are 250 registered patients. Of those, there are 207 approved lab reports. Of those, there are 130 medical history interviews, and then 49 blood samples from patients and 71 other family member blood samples. The graphs in the middle are from their last quarterly report, and this is just their synopsis of the data they're seeing over time. It's an ongoing study, and every quarter they show us kind of what they're seeing with these different phenotypes. If you haven't already, we encourage you to join this study. It's online, it's offered in 4 different languages with more to come. There's a QR code there and then the link below if you'd like to join if you haven't. And then the genotype-phenotype correlation study was sponsored by the University Medical Center Ljubljana. And as you've heard in other talks, it included 125 CTNNB1 patients across 24 different countries. Data was collected through interviews of parents or guardians. And then there was a sub-study by Dr. Edwin Chan to investigate the effects of the mutations on the beta-catenin function. Okay, and now I'm going to hand it over to Anna, if she's there, who can come to the stage and continue the presentation.
Now, sorry, it's the first time I touched this microphone. Thank you, Emily, and thank you, everyone. So one very important thing that has been generated from all these collaborations and everything that Emily has explained is a number of resources. And all of these resources are also a generation led by these organizations. So I'm going to now explain a bit all of this part of the story of all the research resources generated. But I want to start on where all this come from. Why have we generated all of these resources? Because we need to understand better the disease. Like yesterday was very well explained, the natural history study will help us a lot on understanding better the disease, but you also have ways in research that you can also achieve this with either animal models and others. Why? Because we do not know what mutations cause. Is it a loss of beta-catenin expression? Is beta-catenin being produced, but is this dysfunctional? Or do you get a shorter, truncated version of beta-catenin? And maybe this one, instead of having a good function, is actually interfering with the activity of the few good beta-catenin that your children are producing. To be able to answer all of these questions, we have a series of tools that need to be generated, and these are disease models. Many of you have heard through the conference last year and throughout this conference so far that there have been cells from patients that are derived from there. So you take the blood of the patient, you generate a pool of cells, and from these, like Andrea explained yesterday, for example, you get a two-dimensional culture or a three-dimensional culture of the cells of the patient, and with this you can use them to study the disease to try to understand it better. And on top of that, we have the animal models that have been developed. So what CTNNB1 cells and man models have been generated over the past years thanks to the support of the patient organizations to research. So for the pluripotent cells, we have two sources available. One is the Simon Searchlight, where any researcher that is interested has a number of mutations of cells with different mutations available. They are from the type of nonsense mutations and also from the frame shift mutations. There you have the list. I'm not going to be naming them all one by one because that maybe it's a bit boring for the moment. And then for the CTNNB1 Foundation, sorry, Emily, I still couldn't tell the other one. The CTNNB1 Foundation also has a number of cells available for anyone who might be interested for the research. And on top of the frame shift and the nonsense, there's also one with an intron 7. I must mention also here that, as you can see, one of the mutations is actually available in both sources, for example. Now, when we talk about animal models, many animal models have been generated over the past years. We have one whole body loss function model, the same one that Emily mentioned at the beginning. There are 4 beta-catenin deficient models that have been generated by research groups. These ones have different types of deficiencies that are localized at different places of the central nervous systems and others. We have one that is a knockout for neural tube-derived tissues. Another one has knocked out the PV interneurons. Then we have another one with the forebrain excitatory neurons. And then we have, I think you're going to maybe hear about this one a bit later today, the bat face that has mutations on the armadillo repeats. All of this sounds really technical. It is. Even I don't understand it all, but it's important information for any researcher that might be listening to this talk or seeing it in YouTube so that they can see everything that has been generated and everything that is out there. And finally, we have 2 beta-catenin overexpression models, also neural tube overexpression and an APC conditional knockout. And yes, so you have heard about, we are here for the therapies. This is the reason that all of us are here. And now I'm going to show you an overview of all the therapies that are being developed by different programs that are all sponsored by patient organizations. So first of all, we have RNA therapy. You already heard about it last year in the conference, and there are different initiatives from the National Institute of Chemistry here in Slovenia, but also the Tufts University and Johns Hopkins in the United States. Both of these programs are at a preclinical stage, and the target is not to try to fix what is wrong in the cells of our children, but is more to help the allele of our gene that is healthy to produce more beta-catenin to compensate the deficiency. As with every therapeutic, you have heard a lot about it yesterday, every therapeutic has certain considerations. There are no black and white situation. Everything has pros and cons and everything. So the considerations to take into account for RNA therapy are that it's well established for other diseases. There are other diseases already working with this type of therapy, so there is knowledge generated. The dosage can be adjusted and the frequency as the patient grows or reacts to it in one or another way. You can do multiple administrations a long time. So this also allows a lot of flexibility. Also, in case of adverse effects, you can withdraw. But there is, of course, a risk of inconsistent delivery, and there can be immune reactions leading to side effects. I can already tell you that this is generally a quite common one for not only this, but many medications that we take on a day-to-day basis. Then we have the gene replacement therapy, which is the programs that, for example, the one that is being led by the CTNNB1 Foundation together with Children's Medical Research Institute. You have already heard a lot about it today. It's also one of the reasons when we look forward to that clinical trial as soon as possible. As you know, we are in a manufacturing stage. And on top of that, it was presented in last year's conference at the United States that there is also in development a muscle-specific gene replacement therapy program that is right now at the discovery phase. The goal of this type of programs that use the vectors that Andrea presented yesterday is to bring a healthy copy of the gene into the cells of the patient, but this will not integrate with the genetic material of the patient. And as you can see, there are two programs, one that is targeting more at the central nervous system, the one that we are developing at the moment for next year. But the one that is muscle specific is targeting more muscle problems locally. As with the program for RNA therapy, this is well established for other diseases. We saw many examples yesterday of many approvals in the last years. AAV9s, which are the base of these two programs, are the safest tested viral vector. The dosage is not adjustable, meaning that once you get that dose, you normally cannot have a repeated administration, so you cannot have an adjustment. Although science is also working very fast to try to work around this to be able to make it happen, but not as of today. Single administration, like we already heard in the past day, and there is, as usual, a risk of immune rejection, inflammation, and genotoxicity. Then we move into another type of molecules that are the small molecules. And after this talk, you're going to hear about these two programs by the persons that are leading them. Tufts, together with the Broad Institute, are developing a GSK3 inhibitor, and the Italian Institute of Technology is developing a beta-catenin and N-catenin stabilizer. They are both either at preclinical or discovery phase, so still early phase of development, and they have different objectives. The GSK3 inhibitor is trying to increase the beta-catenin levels by avoiding that is degraded in our cells. Every cell in our body has a complex called the beta-catenin destruction complex, and the job is to distract it. So if we can avoid that complex to distract so much beta-catenin, we can also compensate the lack of beta-catenin that we have from the other allele. The approach from the Italian colleagues is different, is trying to enrich the few beta-catenin that is being produced in the bodies of our children, try to enrich it in the synapses. The synapses are the connections between the neuronals, the cells in our central nervous system. So if we can enrich the pool there, maybe we will improve those connections, and that could do something on our neuromotor development for the patients. These programs are also well established for other diseases. Small molecules are very common in the pharmaceutical industries. They're adjustable, again, like RNA therapy in dose and frequency. They can also be used in multiple administration, but you have the potential of the risk of potential off-target effects that can also lead to adverse effects. And finally, there is yet another therapeutic approach that is being studied, which is the DNA therapy. You have maybe heard a lot of it on the news because also very recently the first CRISPR-Cas type of gene therapy has been approved for complete other diseases. So this is still in discovery phase, and this is really a gene editing tool. The same way that I say that in gene replacement therapy, what you introduce in the patient doesn't integrate with the material of the patient. In this case, yes, we are cutting, and we are cutting what is wrong, and we are pasting something that is good. So as you can imagine, this has another level of complexity. In comparison with the other approaches that I have just presented, this is a promising innovative technology, but is not yet established. The long-term effects are still very unclear because this is an integrating technology. We are really touching the the genes of the patient, and there are a risk of unintended changes in the genome. And with this, we have provided you with a whole overview of, I hope that this slide, although it seems really busy, it can really showcase all the efforts, all the work, all the time that is invested, all the fundraising that has to be invested to make this happen. You can see that we are not talking only about 1 or 2 or 3 approaches, but the whole community is working really hard in many approaches, all of them with advantages and disadvantages. And we have examples that have reached really, really far advancement. Now we are in a manufacturing phase. And this is just a great example of when communities and parents work hard and work together, how much can be achieved. So I would like to also thank Emily for the great collaboration preparing these slides. And Emily, you want to say some final words?
Oh, so sorry. Yes, thank you guys. Anna, thank you. It's been a pleasure. And we'd also like to thank all of the organizations listed or not, all of the researchers listed or not, every partner, everyone who is in this together for better lives for our kiddos.
Yes, thank you all for the hard work.
Woo! Thanks, Emily. Thanks, Anna. That was a great download of all of our initiatives. I appreciate it.